Somewhere in the DNA of the average African American, there is a pattern that should not exist by accident. It shows up on one chromosome more than any other. It shows up in the Y chromosome lineages passed from father to son. It shows up in the mitochondrial DNA passed from mother to child. And when geneticists lined all of it up, they found something the historical record alone could never have proven with this level of precision. A signature written by millions of individual, mostly unrecorded decisions, made over more than 3 centuries, by people who never knew their choices would still be readable in the genome of a stranger 400 years later. This video is not about proving one group superior to another. Genetics has settled that question decades ago, and we'll get to why. This is about something stranger, and frankly, more interesting. The fact that 37 million Americans who identify as black and the roughly 200 million who identify as white are not two separate genetic populations quietly living side by side. Their DNA is entangled, asymmetrically, non-randomly, in a pattern that maps almost perfectly onto power, coercion, and the specific brutal mechanics of American slavery. Let's start with what the framing gets wrong before we get to what the DNA actually reveals. For most of American history, black and white were treated as if they described two fixed, separate biological categories, a line you either stood on one side of or the other. States wrote laws defining exactly how much African ancestry disqualified a person from legal whiteness. Virginia used 1/16. Other states used the so-called one-drop rule, treating any known African ancestry, however small, as enough to make someone legally black. These weren't scientific categories. They were legal and social ones, built to control who could own property, who could vote, who could marry whom, who could be enslaved. The question geneticists eventually asked was simple. If you strip away the law and the social category and just look at the genome itself, what do you actually find? To answer that, you need to understand what ancestry even means at the level of DNA. Every person inherits roughly half their autosomal DNA, the DNA on the 22 non-sex chromosomes, from each parent. Trace that back far enough, and you can estimate, statistically, what proportion of someone's genome traces to ancestral populations in different parts of the world by comparing long stretches of shared genetic variation to reference populations from those regions. This is called admixture analysis, and it doesn't tell you who your ancestors were as individuals. It tells you the proportional mixture, generation after generation, that produced the genome you carry today. Alongside autosomal DNA, researchers also look at two much smaller, much more revealing pieces of the genome. Mitochondrial DNA passes almost unchanged from mother to child, generation after generation, tracing a single unbroken maternal line. The Y chromosome passes from father to son, tracing a single unbroken paternal line. Neither one tells you about most of your ancestors. They're just two threads out of thousands. But because they follow strictly maternal or strictly paternal lines, they can reveal something autosomal DNA alone cannot. Whether ancestry entered a population's history disproportionately through mothers or disproportionately through fathers. That distinction turned out to matter enormously. In 2010, a research team led by geneticist Katarzyna Bryce, publishing in the Proceedings of the National Academy of Sciences, analyzed genome-wide data from African Americans alongside individuals from West Africa and Europe. Their finding on the median African American genome, roughly 18.5% European ancestry, with enormous variation between individuals, some carrying almost none, others carrying far more. But the detail that stood out wasn't the average, it was the X chromosome. Men have one X chromosome inherited from their mother. Women have two, one from each parent. Because of that asymmetry, the X chromosome carries a subtly different signature of ancestry than the rest of the genome. And in African American genomes, the X chromosome showed elevated African ancestry compared to the autosomal average. The researchers describe this as consistent with a sex-biased pattern of gene flow, an excess of European ancestry entering through fathers, and African ancestry persisting through mothers. That result wasn't a fluke, and it wasn't new. A separate study published in Human Genetics in 2007, led by geneticist Jason Lind, had already found the same asymmetry using Y chromosome and mitochondrial DNA directly. Elevated European contributions on the paternal line, and elevated African contributions on the maternal line across African American populations. Two independent lines of evidence using two different methods, pointing to the same conclusion. A decade later, a much larger study put an actual number on that imbalance. In 2020, population geneticist Stephen Micheletti and a team at 23andMe, working with historians, combined genetic data from tens of thousands of research participants with historical shipping records of the transatlantic slave trade and published their results in the American Journal of Human Genetics. Their calculation: in the present-day United States, enslaved African women contributed to the modern gene pool at roughly 1.5 times the rate of enslaved African men, even though men made up more than 60% of the people forcibly transported across the Atlantic. Meanwhile, European men contributed to that same gene pool at roughly 3 times the rate of European women. In parts of Latin America and the Caribbean, the female bias was far more extreme still. In some regions, the team's estimates put it at more than 10 African women contributing for every African man. Still, the researchers were direct about why. The imbalance is not explained by chance or by voluntary relationships. It reflects, in their words, the scale of sexual exploitation and coercion enslaved women were subjected to, combined with the simple brutal fact that a large share of enslaved men never survived to father children at all. Geneticist Joanna Mountain, one of the study's senior authors, called the female bias particularly shocking, precisely because it runs in the opposite direction from the raw numbers of who was actually enslaved. Here is where the DNA stops being an abstract statistic and starts being a historical document. That asymmetry is not what you'd expect from voluntary intermarriage between equals. It is exactly the pattern you'd expect from a system in which enslaved black women had no legal power to refuse the sexual advances of white men. Plantation owners, overseers, other white men with authority over them, while enslaved black men had essentially no comparable access, legal or social, to white women. Historians have documented this reality extensively through plantation records, court cases, and the testimony of formerly enslaved people themselves. What the genome adds is a second, independent form of evidence, written not in ink, but in inherited biology, confirming a pattern of coercion at a population-wide scale that no single historical document could capture on its own. This is the moment the story usually gets flattened into something simpler than it is. So let's slow down, because the next layer complicates the picture further. Before we get to the European side of the equation, there's another question. The same 2020 Micheletti study set out to answer exactly which parts of Africa is that African ancestry actually from, historical shipping manifests compiled by historians into a database of tens of thousands of individual voyages, record where enslaved people were forced aboard ships along the African coast. Senegambia, Sierra Leone, the Gold Coast, the Bight of Benin, the Bight of Biafra, and West Central Africa, the region spanning present-day Angola and the Democratic Republic of Congo. The Micheletti team compared that historical paper trail against genetic segments shared between people in the Americas and people from those same African regions today. And, for the most part, the two records lined up. The strongest genetic connection to Americans of African descent traces to West Central Africa, the single largest source region in the entire slave trade, from which historical records show an estimated 5.7 million people were forcibly transported. That matches the paper trail closely. But then, there was one discordance the researchers couldn't ignore. Nigerian ancestry, genetic signal tracing to the Bight of Biafra, the region around present-day Nigeria, showed up in American genomes far more often than the original transatlantic shipping records to the United States could explain. Shipping manifests simply didn't record enough voyages arriving directly from that region into North America to account for how much of that ancestry researchers were actually finding. For a moment, the genetic data and the historical data seemed to contradict each other. The explanation turned out to be a second, less discussed slave trade hiding inside the first one. Between 1619 and 1807, enslaved people were also traded within the Americas, sold, resold, and forcibly relocated between colonies, particularly out of the British Caribbean, long after their initial voyage across the Atlantic had already ended. Historians have only recently been able to reconstruct the scale of this intra-American trade from colonial port records, newspapers, and merchant accounts. Once the Micheletti team factored that internal trade back into the model, the discordance resolved. People with Bight of Biafra ancestry weren't missing from the historical record. They were arriving in North America by a second, indirect route the original transatlantic manifests alone could never fully capture. It's a small technical correction with an unsettling implication. Even the paper trail of the slave trade, as thorough as historians have made it, undercounts how many times a single human being could be bought, sold, and moved again. If European ancestry entered the African American gene pool primarily through non-consensual and coercive circumstances during slavery, you would expect that ancestry to be concentrated most heavily in the States, where slavery was most entrenched, longest running, and least restrained by outside scrutiny. And that is exactly the geographic pattern researchers found, but not in the way you might guess. The 2015 study by Bryce and colleagues, drawing on genetic data from over 5,000 self-identified African Americans across the country, found that African ancestry proportions were actually highest, not lowest, in the Deep South, particularly South Carolina and Georgia, and progressively lower moving toward the Northeast, the Midwest, and the Pacific states. On average, the study estimated African American genomes at 73.2% African ancestry, 24.0% European ancestry, and 0.8% Native American ancestry nationwide, though those numbers shifted meaningfully by region and, again, varied enormously between individuals. Why would the states with the deepest, most brutal history of slavery show the least European admixture, not the most? A separate and even larger study helps explain it. Published in PLOS Genetics in 2016, a team led by geneticist Sohail Baharian analyzed genomic data from nearly 3,800 African Americans and modeled the demographic history behind the numbers. Their estimate, on average, about 82% of the ancestors of today's African Americans lived in Africa before transatlantic travel began. Roughly 17% lived in Europe, and just over 1% lived in the Americas already as indigenous peoples. And critically, their models indicated that most of the admixture between African and European ancestors happened in the South before the Civil War, concentrated in the era of slavery itself, not afterward. So if the mixing happened mostly in the South, why does the South show the highest African ancestry today rather than the highest European ancestry? The answer runs through one of the largest internal migrations in American history, the Great Migration. Between roughly 1910 and 1970, millions of Black Americans left the rural South for cities in the North, Midwest, and West. The Baharian study found that this migration wasn't demographically random. It reshaped the genetic relatedness map of the entire African American population, and it appears to have been ancestry biased, meaning the populations that moved and the populations that stayed behind were not identical in their ancestry profiles. Combined with additional, smaller waves of more recent African immigration concentrated in some southern communities, the result is a present-day map that looks almost counterintuitive until you understand the two forces layered on top of each other. Historical admixture concentrated in the plantation South, followed by a century of internal migration that redistributed the population without erasing the original signature. But there's a second half to this story, and it's the half almost nobody brings up, because it complicates the Black and white America framing even further. That same 2015 study found that European Americans are not a genetically closed category either. Nearly 4% of self-identified European Americans in the study carry detectable African ancestry, a small percentage nationally, but not statistically negligible, and again concentrated more heavily in certain southern states than others. Some of that reflects the well-documented historical phenomenon of individuals with partial African ancestry, who, over generations, were categorized or self-identified as white, sometimes called passing, as they moved, married, or as social boundaries shifted around them. The genome doesn't forget that history, even when the historical record does. Put both halves together and you get the actual finding, not the sensational one, the real one. There is no clean genetic line separating Black American and white American as biologically distinct populations. What exists instead is a single, continuous, unevenly mixed American population, produced by centuries of forced migration, coerced reproduction, geographic sorting, and voluntary movement, with a strong and measurable directional signature left behind by the specific power imbalance of slavery. The DNA doesn't just show that mixing happened. It shows, with a precision no diary or plantation ledger could match, how it happened, through which parent, in which generation, concentrated in which region. It's worth being precise here about what this evidence can and cannot tell you, because overclaiming would betray the whole point of using science in the first place. Genome-wide admixture estimates are population-level statistical inferences, not individual family trees. They can't identify specific ancestors, specific relationships, or specific acts. Trees, different studies, using different methods and different reference panels, have produced somewhat different average percentages over the years, and researchers themselves have debated why some estimates run higher or lower than others. The precise timing of when admixture peaked, how much of it happened during slavery versus in the decades immediately after, and how consistent the sex bias pattern is across every region and every era, are still active areas of research, not settled to the decimal point. What is well supported, across multiple independent studies using different techniques, is the overall shape of the pattern. Substantial, geographically variable African and European admixture in the African American population, a genuine sex bias favoring European paternal and African maternal contributions, and a small but real African ancestry component within the self-identified European American population. This history isn't only preserved in the genome. In at least one striking case, it's still actively shaping health outcomes today. African Americans develop chronic kidney disease at substantially higher rates than European Americans, and for years, the reason was unclear. In 2010, geneticist Giulio Genovese and colleagues, publishing in Science, used exactly the kind of admixture mapping this video has been describing, scanning the genome for regions where African ancestry correlated with disease risk, and traced the excess risk to two specific variants in a gene called APOL1, located on chromosome 22. These variants are common on African chromosomes and essentially absent from European ones. Follow-up research found that having one or two copies of these variants accounts for a large share, later estimates put it at around 70%, of the excess kidney disease risk in African Americans, and today an estimated 13% of African Americans carry two copies of the risk variants, the combination associated with the highest risk. Here's the twist that turns this from a disease statistic into a genuine evolutionary story. Those same variants aren't simply bad. In West Africa, they do something useful. They help the body kill Trypanosoma brucei, the parasite that causes African sleeping sickness, a disease that has killed people across the region for millennia. Natural selection favored these variants in West African populations because the short-term survival advantage against a lethal parasite outweighed the long-term cost, a kidney disease risk that, historically, would only appear decades later, often after people had already had children. It's the same evolutionary trade-off that produced the sickle cell trait's relationship with malaria resistance, a genetic variant that helps in one environment and harms in another. And because of the transatlantic slave trade, a variant that evolved as protection against a parasite in West Africa is now, generations later, and an ocean away, a measurable driver of kidney disease risk in American hospitals. The same admixture that this video has been tracing through the X chromosome and the Y chromosome and the mitochondrial line isn't just a historical curiosity. Geneticists use it today, directly, to find disease genes that would otherwise be invisible. There's a deeper layer underneath all of this, and it's the one that tends to surprise people most, because it reframes the entire premise of black versus white at the level of the species, not just the country. Modern human genetics has established, through decades of population genetic and paleogenomic research, that all present-day humans descend from populations that originated in Africa, with the greatest human genetic diversity on Earth found within Africa itself, not between Africa and everywhere else. Sarah Tishkoff and colleagues, publishing in Science in 2009, mapped the population genetic structure of Africa in unprecedented detail, and showed that African populations alone contain more genetic variation among themselves than exists between many non-African populations combined. The categories, African and European, as used in these ancestry studies, are not fixed biological castes. They're statistical shorthand for genetically diverse, historically connected human populations that themselves emerged from thousands of years of migration, contact, and mixing long before anyone crossed the Atlantic. Race, as a rigid biological hierarchy, has no support in this data. What the data does support is that populations moved, mixed, and diversified constantly, and that the American chapter of that story, however specific and however painful, is one more layer on top of a much older and much larger pattern that includes every human being alive today. So what does it actually mean then when a DNA test tells an African American client they carry 20 or 30 or 40% European ancestry? It doesn't mean they are part white in the identity sense that phrase implies. It means that somewhere in the last 400 years, in a specific place, under a specific and often violent set of historical circumstances, ancestors of theirs, disproportionately fathers rather than mothers, were European. And it means that same signature, read carefully, is one of the clearest genetic records we have of exactly how coercive, non-consensual, and asymmetrical the sexual politics of American slavery actually were, preserved not in a single document that could be lost or destroyed, but distributed across the genomes of tens of millions of living descendants. That's the part that tends to get lost in the sensational framing. This was never a story about two separate bloodlines finally being compared. It's the story of one entangled population whose genome preserves, with startling fidelity, the exact shape of the power structure that produced it, visible on the X chromosome, visible in the Y chromosome and mitochondrial lineages, visible in the regional map of who stayed and who left, and visible in the small but real African ancestry, hiding inside millions of genomes currently labeled on paper as simply white. The DNA was never trying to keep a secret. It just took modern genomics long enough to learn how to read what was already there.